The ChIP experiment is used to monitor dynamic changes in histone modifications that occur during STAT1 activated gene expression. Alterations in the occupancy of transcription factors, histone modifying complexes, and the transcription machinery itself can also be assayed in this way. To begin the assay, cells are induced with interferon gamma to activate STAT1 signaling. Cross-linking the cells with formaldehyde and collecting them follows. Proceed to isolate nuclei and sonicate, which shears the chromatin into approximately 200 to 1000 base pair fragments. Next, add an antibody that targets the protein or histone modification of interest, and then precipitate the immunocomplexes. The immunocomplexes are washed, the cross-links are reversed, and the DNA is purified. The changes in histone modifications or recruitment of proteins to specific areas of a gene can be determined based on quantitative real-time PCR analysis of the immunoprecipitated DNA.
Hi, my name is Lauren Buro from the laboratory of Dr. Melissa Henriksen in the Department of Biology at the University of Virginia. Today we're going to show you a procedure for chromatin immunoprecipitation. We use this procedure in our lab to study dynamic histone modifications that occur during transcription downstream of STAT1 signaling. So let's get started.
One day before beginning this procedure, culture the cells so that a sufficient number is available. For the 2 FTGH cells used here, prepare one 15 cm tissue culture dish at 80% confluency for each ChIP assay. In each experiment include a negative IgG control, a positive pan-histone antibody control, and duplicates.